The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 33 of the eLife podcast, which is produced by The Naked Scientists. I'm Chris Smith. This month, the sleeping sickness bug is found lurking in the skin, the genetic trick that protects elephants from cancer, and what's life like for a scientist who's a woman. But first, the heart is one of the first organs to form during mammalian development, and in a human, it was thought to form a beating structure from about 3 weeks following conception. But now, new imaging techniques have shown that the process occurs even earlier. By just 16 days, there are already cells destined to form the future heart. These cells show surges of calcium that will later translate into heartbeats. The discovery is important because, with 1 in every 180 babies affected, congenital heart disease is one of the most common developmental disorders. The discovery may also inform how we progress the use of stem cells to repair damaged adult hearts too. From the University of Oxford, Paul Riley.
Basically, we found that the heart beats earlier than was previously thought. So it starts to beat with very early forming heart muscle cells as the heart forms a crescent shape. And this was before the stage that was previously thought, which was more like a linear heart tube, which is in the mouse at least a sort of day and a half later, which equates to humans of about 5 days time difference.
So in a human embryo, how early would we, if humans are analogous to the mice you've been studying, begin to see a heartbeat?
So if the analogy is exactly accurate, and you have to take that with a slight grain of salt, the match-up would be around about day 16 of human pregnancy.
Which is incredibly early, isn't it? I mean, there's not very much to a human embryo at that stage of development.
That's correct. And certainly the heart, in terms of the sort of organ we would perceive, is a very early forming tissue, and it really hasn't got all of the structure that we would normally associate with the heart at that stage.
How does the heart form?
Well, it forms for a whole series of cellular and molecular processes, but basically there is this specification of cells either side of the embryo's midline. They come together at the middle of the embryo, they form this linear heart tube. That heart tube loops, and this looping aligns the chambers correctly and the inflow and outflow regions of the heart. Those chambers then specify and grow. They divide to make sure the blood flow is in one direction, and then there's further growth and expansion. So that's in a simplistic way of looking at how the heart forms. So we're talking about the very early stage when the heart exists as a few cells either side of the embryo's midline.
And how did you find that these heart cells are effectively beginning to beat so early in development, and why was it missed before?
Well, I think the issue here is the work that Shankar Srinivas has sort of pioneered in imaging the live early mouse embryo. And so what we were able to do is we were able to look at very, very early stages and then appreciate that these cells were actually contracting. And then we were also able to look at the key events that are required for that contraction. How do these heart cells beat, and what's the requirement for that? And it turned out to be calcium signals.
Why is calcium important?
Calcium is very important because it interacts with the contractile machinery of the heart muscle cell, and that enables the contractile process, so the beating. But what we found was actually cells were positive, if you like, for a calcium signal before even the contraction event occurred.
Right, so are you saying then that these cells already know that they're going to be cardiac cells? They're already beginning to set up these fluxes or spikes of calcium, which would normally coincide with a heartbeat, but that then comes along a bit later. So the calcium spikes might be in some way encouraging the cells to turn into heart cells.
Absolutely correct. The idea is that we think the calcium signal is an early defining event. It happens in a sort of random process within cells, and we don't understand how that occurs and which cells tend to turn on the signal before others, and also how those cells then synchronize this calcium signal. But what we think happens is the calcium then is also important for the actual process of this heart muscle cell differentiating and maturing properly.
And given how common the congenital heart problems are as a group, I suppose understanding this process and how heart cells get specified is going to be fundamental to understanding congenital heart disease, but also potentially putting hearts right.
That's correct. We think that the twofold application of the work if you extend it into sort of the human setting and the disease setting. And that is one, understanding the fundamental processes of birth defects and congenital heart disease, i.e. at the level of how the heart starts to contract, what happens if that goes wrong, if, for example, those cells that start to beat don't synchronize properly and don't communicate with each other properly. And we also think there's an important component here as applies to adult heart repair, because if we're thinking about introducing new heart muscle into a patient who's had a heart attack, for example, then how that heart muscle needs to start to beat and integrate and communicate with the survived heart muscle of the patient is really important.
And do you know yet what those cells are doing in order to produce these interesting calcium signals and how they're doing that?
Well, we know that one of the key events is they need to express a particular ion channel, which actually exchanges the calcium for sodium. And interestingly, so there's a sort of an ionic balance of these different signals. And so the expression of this channel, which is called NCX, so the sodium calcium exchanger, is really important in these specifying heart muscle cells at these early stages. And what that enables then is for calcium to enter the cell. And so that's a key event. We don't know why, as I say, particular cells in this early forming heart start to do that earlier than others, and the progression of events and how that becomes sort of synchronized so that these heart cells communicate. And these are studies which are ongoing, particularly in my colleague Shankar Srinivas's lab, as he looks at single cell sequencing to determine key molecular profiles of these individual cells from this forming heart.
Paul Riley on when the first of a lifetime's 3 billion heartbeats begins. African sleeping sickness, or trypanosomiasis, is one of a list of tropical diseases that the World Health Organization wants to stamp out, and they've set an ambitious target to cut cases to under 1 in 10,000 of the population in 90% of the areas that are affected. But new research from Glasgow parasitologist Annette MacLeod suggests that there might be a tsetse fly or two in the ointment, because she's discovered a formerly unrecognised reservoir for the infection, lurking in the skin of apparently healthy people.
African trypanosomes are protozoan parasites that cause the fatal disease human African trypanosomiasis, or sometimes known as African sleeping sickness. And this is found in sub-Saharan Africa, and it is transmitted from person to person by the bite of a tsetse fly when it takes a blood meal. And the number of cases of sleeping sickness has gone down in recent years due to concerted efforts by the WHO and national control programs to try and control the disease. And the number of cases has dropped below 10,000 for the first time in 50 years. But we've been here before. In the 1960s, the number of reported cases was below 10,000. And then we took our eye off the ball and the disease re-emerged, and it peaked to about 300,000 in the 1990s. Now we have the disease under control again, and the important thing is not to let the disease re-emerge. But there are still these stubborn pockets of disease that just won't go away. And we need to understand how the parasite has escaped the elimination before so we can stop it doing it again.
Where do you think it might be lurking?
Well, we think there are some people that have low numbers of parasites in their blood and they have no symptoms. And these people are missed by the screening process, but they are infected, and evidence suggests that they are able to transmit the disease. And the question is how?
I guess then, so where are the parasites lurking in those people? If they are not in the blood, where are they?
That's a crucial question, and that's what we have discovered. We have discovered a hidden reservoir of these parasites hiding in the skin.
In the skin?
In the skin. These skin-dwelling parasites can be picked up when the tsetse fly takes a blood meal and so spreads the disease.
I guess everyone had assumed that because these insects are drinking blood, that must be the crucial route through which anything gets transmitted into the fly. But if the parasites are in the skin, in the course of getting to the blood, that's when they might be picking them up.
Yes, because tsetse flies have serrations along their proboscis that actually makes a mess of the skin, and the tsetse bite is quite sore, and it feeds on the pool of blood and lymph that collects as it bites.
People talk about this being slash and suck, don't they?
Yes, that's right. It's a slash and suck kind of insect.
And so if these trypanosomes are sitting there in the skin as this serrated mouth part goes through, then it could draw in the parasites via that route.
That's right.
How do you know this is happening? How did you find this?
Well, what we did, we had fairly simple experiments where we labelled the parasites, and then we used different live in vivo imaging technologies to observe where the parasites go.
When you say label, how?
We have them expressing a fluorescent tag so that they glow red or green. And what we found is that we could follow these parasites in a live animal over time.
And do you think this is relevant to what's happening in these people? Because obviously you're not doing this in people, you're doing this in experimental animals. So do you think that's a good representation of the human equivalent?
Well, yes. So first of all, we looked in mice and we found that the parasites were in the skin of mice. And we found that these skin-dwelling parasites in mice could be transmitted to tsetse flies when we fed tsetse flies on the mice. And some mice have very low parasitemia, so that means that they had very low numbers of parasites in their blood. But they did have skin-dwelling parasites. And these skin-dwelling parasites were able to infect the tsetse flies. Obviously, you can't do these experiments in humans, but we managed to obtain skin biopsies for over 1,000 people living in an endemic focus in the Democratic Republic of Congo. And we looked in these biopsies for the parasites. And we found people that had parasites in their skin. And these people had not been diagnosed before. They had not been diagnosed as having sleeping sickness. They didn't have clinical signs, and they didn't seem to have any parasites in their blood.
But, crucially, does the bug respond to the same treatment one would mete out to somebody who has parasites in their blood or in the skin? In other words, if we can find it, we can just get rid of it.
That's something that hasn't been tested before. We've not tested the current drugs to see if they can eliminate these skin-dwelling parasites, but that is a crucial step that we must do. And if any new drugs come along, we must make sure that they are able to kill these skin-dwelling parasites as well as the blood parasites. So current policy is not to treat people with asymptomatic infections, but we think that needs to be reconsidered in the light of these individuals that can persist and transmit the disease.
And Annette also told me about a Sierra Leonean man who'd lived in the UK for 29 years and only developed the symptoms of trypanosomiasis, presumably picked up many years previously after he received immunosuppressive therapy for a totally different illness. This presumably brought the disease out from hiding. You're listening to the eLife podcast with me, Chris Smith. And still to come, where are all the women who start off in science? Plus, what the brain makes of the rubber hand illusion. But first, it follows logically that the bigger you are, the more cells you have, and therefore the higher the likelihood of one of those cells becoming cancerous. But if you look at progressively larger animals, you don't see an increased rate of cancer. This is called Peto's paradox, after Oxford University's Richard Peto, who pointed out on a cell-by-cell basis, humans have a lower cancer risk than a mouse. So why does it happen? The University of Chicago's Vinnie Lynch has been looking to elephants for inspiration.
So we were curious in why elephants don't get cancer, and we cared about elephants because their closest living relative is actually very, very small. It's about the size of a groundhog or a small cat or something. It's called a hyrax. So within a few million years after these things diverged, elephants ancestrally went from being very small to being huge. So because these two things are closely related to each other, we can therefore compare their genomes to see what genetic changes occurred in elephants that might be associated with an increased ability to kill cancer cells or maybe just stop cells from becoming cancerous to begin with. So we basically just looked in the elephant genome for things that might be associated with cancer resistance.
Did you just go to the database and get the DNA sequences of different elephants, or did you get some real elephant DNA for yourself?
So we have real elephant DNA and we have elephant cells, but it was even easier. So for 10 years or more now, the NIH and lots of other foundations around the world have been sequencing animal genomes. So the elephant genome and the hyrax genome, along with many others, had already been sequenced. So we were basically able to just go to this website and type in a bunch of the names of the genes that we were interested in and see them pop up on the screen. So we did that for a couple elephant genes and found some things that we thought might be associated with enhanced resistance to cancer in elephants.
Now, did you literally do a letter-by-letter comparison, or were you saying, well, we know that there are certain mechanisms that cells use to stop cancers in their tracks. There are certain genes that cause cancers, so we're going to focus our attention on both of those and see if there's a difference.
Yeah, that's exactly what we did. So, cancer has been very well studied for decades now. So we know that there are basically, at least in cancer biology, these two different kinds of genes. There are genes called oncogenes, and when they're mutated, they cause cancer. And there are genes that are called tumor suppressors, and their job is to suppress the formation of tumors. And they've been very well characterized. There are about a few hundred of each kind. So we just went into the elephant genome and counted up how many tumor suppressors they had and how many oncogenes they had. And we found that for this one special kind of tumor suppressor, they had 20 copies, whereas every other animal that we looked at had one.
Are those copies all functional? Because one of the things that we have learned from the Human Genome Project is that there are sort of fossilized copies of certain genes lurking in the genome. And when you read for them, you can find these genes there. Doesn't mean they work, though. So do these 20 copies in the elephants actually do something?
So it looks like about half of them. So maybe 10 have signatures that they do something, and then the remainder have signatures of these non-functional genes. They're called pseudogenes because they look like genes, but they're not really genes. So we think a subset of them are actually functional.
And the oncogenes, did you see any evidence that they have fewer abilities for cells to become cancerous in the first place?
So we didn't find that elephants had fewer oncogenes, right? So that'd be the strategy. If you have these genes that when they get mutated cause cancer, one way you can reduce your risk of developing cancer is to just get rid of these genes. But elephants had just as many as everybody else. And that's not entirely unusual because these sort of oncogenes play essential roles in animal development. So it's probably the case that if you got rid of them, your development would be weird. So they're probably really constrained. You can't mess with them.
So it does look like the mechanism, at least in elephants, is a big expansion of your ability to suppress cancer in cells. But what about other animals? Because elephants, just because you see it in them, it doesn't prove the rule, does it? What about other close relatives of elephants, things like mammoths or other massive animals like whales? Have you looked in those examples, for instance?
So we've looked in mammoths, and mammoths are most closely related to Asian elephants, and both mammoths and Asian elephants, for which there's also genome data, have similar numbers to African elephants. So there seems to be this increase in the copy number of this special gene called TP53 seems to be an elephant thing. But then we did obviously figure if it's expanded in elephants, what's going on with these other things that don't seem to get cancer at the rate that they should. And at least for whales, we didn't find a change in the number of this gene in whales. So whales have one copy just like everybody else. We did, however, see that this one lineage of bats, which are very small but live a very, very long time, they may have extra copies. We haven't tracked it down entirely yet, and we're working on that now. But it looks like, at least initially, that they have one extra copy.
And rhinos, hippos, they're also pretty big. Do they also show an expansion of these P53 genes?
So we've looked at everything that we could find, including rhinos and hippos, and they don't seem to have an expansion of these P53 genes, which is probably telling us something more fundamental about evolution, in that there's more than one way to solve a problem, right? So elephants found this particular way to solve the problem, but the other lineages that get big or live a long time, like bats that live a long time or whales that live a long time, and rhinos and hippos, they just found other ways to do it.
Vinnie Lynch. If you're feeling brave enough, you could try this. Lay one hand on a tabletop and put the other out of sight below. Put a rubber hand on the table where your hidden hand would naturally rest next to its neighbour. Have someone stroke the index finger of your real hidden hand and the index finger of the rubber hand at the same time. It's very hard to resist forming the distinct impression that the rubber hand in front of you isn't your own real hand. And if someone actually comes along and hits it with a hammer, you feel momentarily terrified that it's going to be agonisingly painful. Why this rubber hand illusion works, we don't know, but Francesca Gabarini has found that when the illusion takes place, the motor circuits controlling movements in your real hand become very powerfully depressed, as though they've been deprioritised as your attention transfers itself to the fake hand and you assimilate it into your body image.
The sense of body ownership is the feeling that the specific body parts belong to ourselves. that we usually take for granted, but it can be selectively altered after brain damage when patients lose the capacity to discriminate between the own and the other body parts, or sense of body ownership can be also altered during a famous experimental procedure that is the rubber hand illusion.
Okay, so tell us first of all, for those not in the know, what is the rubber hand illusion and how do you do that?
Watching a rubber hand being touched while one's own hand, hidden of the view, is touched synchronously leads to a sense of body ownership over the rubber hand. And also this leads to a shift in the perceived position of the real hand towards the rubber hand. And this generates a sort of disembodiment in the real hand of the subject.
So in the mind of the person who is seeing a rubber hand on the table in front of them, their own hand might be, say, hidden under the table, and you stroke the rubber hand in the same place as you stroke their real hand, you end up with the person believing that the rubber hand is their own hand, and one would believe that their body image, the brain accommodates their body image to include the rubber hand rather than their own real hand.
Yeah, we are able to manipulate the sense of body ownership in healthy participants, but it's important to keep in mind that healthy subjects are always aware that it is an illusory procedure, so they are not inducing thinking that the rubber hand is actually their own hand.
And once you've got the people experiencing the rubber hand illusion, what measurements do you make? What do you do to them next?
Okay, we are interested in recording some motor parameters because we are interested in investigating how the human motor system reacts when subject's sense of body ownership is altered during the rubber hand illusion. So during the experiment, the region of the brain that controls movement, and in particular hand movements, was artificially stimulated by using a specific technique that is transcranial magnetic stimulation, TMS, to stimulate the part of the brain involved in controlling movement, that is the motor cortex. So the magnetic stimulation generates electrical signals that travel down to the spinal cord and to the muscles, generating muscle contractions that can be measured and that we called motor evoked potential. And we measured the size of this motor evoked potential. And the motor evoked potential reflects the ability of the motor cortex to produce the movement of the hand.
And what do you find then when you compare the level of preparedness of the motor circuits when the person is or isn't experiencing the rubber hand illusion? Is there a difference?
Yes, while subjects experience the rubber hand illusion, we found a strong, very dramatic decrease of the amplitude of the motor evoked potential. And this means that the motor cortex is less able to induce the movement, and the deluded hand is less ready to move. So this means that the motor system during the illusion is downregulated.
Now, why would that happen? Because the person still believes they have a hand and they still potentially may want to move that hand. So why should believing that a rubber hand on the desk, why should that be reflected in a change in the degree of facilitation or preparedness of their motor areas?
Because if the brain no longer considers the hand as a part of the own body, it is not able to exert a control over a body part that is no longer a part of the body. So what we can see is that the sense of body ownership strictly depends on the possibility of movement, because basically if I believe that the hand is mine, I must be ready to use it.
Francesca Gabarini, she's at the University of Turin. Now, in each episode, alongside the research, we also try to delve into other aspects of practicing science. Previously, we've looked at funding, or more accurately, lack of it, and we've also recently explored public engagement and developing the problem-solving skills of undergraduates. This month, we've a personal insight into a perennial problem.
My name is Indira Raman. I'm in the Department of Neurobiology at Northwestern University.
What really struck me about your perspective in eLife this month, Indira, is that you opened by saying, in my 18 years as a professor of neurobiology, I've been asked to speak as a scientist because I'm a woman, but this is the first time I've been asked to speak as a woman because I'm a scientist. Elaborate.
Well, as one knows, very often in many scientific venues, there's a growing interest in ensuring that the group of people that is speaking represents a diverse cross-section of the scientific population. And so I've been asked to speak as a scientist because of my science, but very often I'm also aware that the organizers are interested in having a representation of women, and I am a woman. But in this case, the focus was different, and I was asked to speak about my experiences as a scientist because of being female. And I chose to do it in a way that seemed to be compatible to me with my experience of talking about science.
Now, how does this whole kind of, I get asked to talk about science because I'm a woman, how does that go down with you? It's good to be gender neutral, but what I think would really upset someone is if they heard that and then thought they had been elected to appear on a programme because of their sex, not because they're good at science.
Right. I think that this is a central question of our time, because on the one hand, we want to encourage diversity, and I think that that's a laudable goal that I support. But we also want to maintain, as you're saying, the highest caliber of participants. I think those things sometimes work together and sometimes move into conflict. And that's why I called it out at the beginning. And that's why I mentioned in the piece the way that I deal with that is internal, with this idea that I walk on stage as a woman and walk off stage as a scientist.
In your view, why are we in the position we're in, where we actually see that at the bottom end of the scientific tree, coming in, the input pipe, if you like, has a very healthy proportion of women? But when you look at what comes out at the other end and turns into people of your sort of calibre, they've all vanished. You're a very rare species. Why?
I don't think there's a simple answer to that question. There are many reasons why people stay in science. There are many reasons why people may leave it. There's one very straightforward element of being female that can be complicated to tie in with a scientific career, and that's childbearing. There's a tremendous element of uncertainty and a lack of security through the one's 20s and often one's 30s. Some people respond to what they see as a social dynamic within science. It can be very competitive. There is the intensity of the work where one really almost never goes home and leaves it completely behind them. And as it turns out, there are proportionately more women who decide this career is not for me.
But the majority of the reasons you have highlighted apply to men too. But they don't leave in the same proportions or en masse the same way the women do they?
Correct. Right. So for whatever set of reasons, there are more women who decide that those attributes of the field are elements that they don't really want to maintain in their daily lives.
People often talk about there being role models, and the way to solve a lot of this is to have good role models, and this will attract more people of a particular persuasion, gender, background, whatever, into a particular area. Do you go along with that, or do you think actually that you can have as many role models as you like? If the system's broken, as soon as you take the pressure off, it will just relapse back to the broken type it was to start with?
I think that it's important to think about role models in a broad sense and think, what is it actually that you are wanting to model yourself after? My own experience was that I worked for men as I was being trained. They were all white American men, and they were tremendous role models for me because what they modeled for me was a way to do science, a way to be just in one's thinking and careful in one's thinking and humane in one's behavior. And those were the things that I wanted to have modeled. I should say that I am half Indian from India and half Caribbean. And there was nobody like me. It was very unlikely I was going to find a half Caribbean, half Indian female role model who was doing electrophysiology for me to model myself after. And so I found other ways of recognizing pieces of myself in people who didn't happen to share many of my attributes. And I have found it possible to be a role model for students from all over the world, male and female. A big discovery for me when I was an undergraduate was finding out that there were other people at my college from many different countries who actually shared many of the experiences that I had. And so I think that as we learn to expand our notion of role models, we will be able to reach across the divides, because otherwise we'll never change the proportions of people in science, because we'll be operating under the assumption that one can only train someone who is like you.
Indira Rahman. And that's it for this episode. You've been listening to the eLife podcast, previous editions of which you can find for free on eLife's website. That's at elifesciences.org, or you can subscribe on iTunes. This is a Naked Scientists production. I'm Dr. Chris Smith, and you can find out more about what we do on our website at nakedscientists.com. I shall be back with another look inside eLife next month, but until then, thank you for listening and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.